Good morning, class.
Good morning.
As we were told on the other day, we were told to research on the assignment about the light microscope, and this is what I found. My name is Rafael Willigafo, and this is my research. The brief history of the invention of a microscope. The first microscope was invented in the 17th century, 1650, by Hans and Zacharias Janssen. It was a compound microscope which combined two lenses for greater magnification. At the same century, Johannes Kepler, a German astronomer, invented a modern compound microscope with most features of the present-day microscope. In 1665, Robert Hooke used an improved compound microscope to observe cells. Later, Anton van Leeuwenhoek invented a simple microscope with biconvex magnifying lenses used to observe the nucleus and unicellular organisms, e.g. bacteria. Both are light microscopes as they use light to illuminate the specimen under view. In the 20th century, 1938, Ernst Ruska, a German engineer, developed the electron microscope, which enabled much of the internal structures of the cell to be observed. Till date, it is widely used in biology to show more details of the cell. Thank you.
Good morning.
Good morning.
So previously we were looking at how to handle and care for a microscope.
Yes. Make sure the low power objective lens is in position in line with the eyepiece before and after use.
Remember, you put the low power objective lens to click into position with the eyepiece lens. So mnaona nimeplace hivyo. Aya, reason ni gani? Reason ni hii. So we have this part. Hii part inaitwaje?
Course
adjustment knob, and this one is the adjustment knob. So when I use the course adjustment knob, I move the body tube. Not hivyo, up and down over long distances. So in adjustment knob, or in a move body tube over short distances, I'm going to click into position the high power objective lens. Naona kwenye nimeiweka? This is the high power objective lens. It's longer than the rest. So what happens if I use the course adjustment knob nikienda hivyo chini? Angalia. Unajua hapa pia kuna lens nyingine. Unaona lens hapo? Na pia hapa kuna lens nyingine pia. So hii course adjustment knob ukitumia kwenda hivyo chini inaweza crash hiyo part. Unaona? Bado inaweza endelea but imefika mwisho. Then sometimes we place the specimen on the stage with a slide. And the slide is made of glass. So what will be the reason? To prevent, you can also break this one. To prevent crashing the lenses and also crashing the slide yenye inaweza kwa stage. Because umeona the high power objective lens inaweza fika chini. It's longer than the rest. Is that understood, people?
Yes.
When you're using the high power objective lens, you only use the fine adjustment knob. So we're going to look at the parts of the microscope and their functions. So we'll start from the top. What is the function of the eyepiece lens?
Contains lenses which contribute to the magnification of the specimen.
Yes. Now, what else contributes to the magnification of the specimen apart from the eyepiece lens in a light microscope? Objective lenses. When observing the specimen on the stage, I have to observe through the eyepiece lens and I also have to observe through the objective lens. So magnification itakuwa eyepiece lens magnification times the objective lens magnification. So for example, if I'm using the high power, itakuwa times 45 times 10. So itakuwa times what?
50.
Times 450. So the highest magnification we have in this microscope is times 450. Meaning it can enlarge an object 450 times. What is the function of the body tube?
Holds the eyepiece and the revolving nose piece.
Correct. See the revolving nose piece?
Yes.
Mbona inaitwa revolving? Juu inafanya nini?
Revolve.
Ina revolve. Mnaona?
Yes.
Yeah. So what is the function of the revolving nose piece? Ndio hii hapa na mnaona hizi hapa hivi chini. Ina hold nini?
It holds the objective.
Alafu, apart from holding the objective lens, what else does it do? What else does it do?
And able to change from one objective lens to another.
In the stage. And as you can see, it's a flat platform.
This is where we place the specimen.
On the stage, we have the clips. Clips ni za nini?
It's a slide on position.
This is the diaphragm, people. You see? What is the function of the diaphragm?
It regulates the amount of light passed through the condenser to illuminate the specimen.
That is true. Then now we also have the condenser.
Concentrates light on the specimen. Concentrates light onto the skin.
Without the mirror, we cannot have what?
Light.
Light. So what does the mirror do?
Reflects light
Uh-huh.
through the condenser.
Yeah, onto the
Stage.
stage to illuminate the specimen. The function of the base.
Provides firm and stable support.
Mama Lim, ni ya nini hiyo? Ina provide support to the stage, and the body tube. Hebu angalieni unaona nini hapa?
The stage.
The stage na hii side nyingine? The body tube. Course adjustment knob. Hiyo tumesema. What is the function?
It's to move the
Body tube.
body tube high up or low in a high
Over what?
Long
Tunasema
distance.
over long distances. Hiyo.
To bring the specimen to rough focus.
Na fine adjustment knob.
To bring the image into a sharp focus by lowering and rising and lowering the part to
The body
of the body tube over a short distance.
Yeah. Have you grouped yourselves, people? Yeah. Now, all of us have to move to the side benches. Side bench ni ile yenye iko ile side na ingine. So, you have your groups and you have a representative. Si ndio? Akujie microscope. Good afternoon. Good afternoon. All right, so today we are in Biolab 2. And what are we doing today?
How to use a microscope.
So I see you're set on the side benches where there is light passing through the window. The first procedure. You place the microscope on the bench with the stage facing away from you. Correct. So in a face your direction, your window. Turn the low power objective lens until it clicks into position with the eyepiece. Aya, mmemaliza? Yes. Aya, tuonyeshe low power. Low power ni gani? How do you know it's the low power? It's written low power. Yeah, it has the least magnification. Sindio? Ya times ngapi? Times 5. Sindio? And it is the shortest. Naget hiyo? Then tuko na objective lenses zingine gani? Apart from low power.
We have the medium power.
The medium power and the high power. Unazijua na hiyo magnification. Medium power iko na times? Times 10. High power iko na times? Times 45. Number 3. Ensure the diaphragm is fully open. Number 4. Look through the eyepiece with one eye. Meanwhile, adjust the mirror under the stage to ensure that maximum light can pass through. Imewak? Haya, umefanya hebu demonstrate vile umefanya. Nani alikuwa anafanya? Yes, so unachungulia hapo na mwingine mirror. Yeah, so you have to learn how to do it yourself because you're the one who is observing.
I see light and
You see light? Yeah. What is the shape of the light?
circular.
Circular.
Yeah.
Mnaget hivyo? Yes. Aya, kuja hapa. Mtu unawakianga wapi kwa microscope? Mtu mmoja simame hapa. Hebu nisongea kidogo. Hivyo ndivyo unafaa kuwaka ukitumia microscope. Unatumia peke yako. Unaget hivyo? Chungulia kwa eyepiece. Shika mirror. Shika mirror. Ian just mpaka time yenye utaona light iko bright. Light iko the brightest. What shape is it?
It is circular.
Circular. We call that the field of view. We call it what?
The field of view.
Number 5. Place the slide containing the specimen on the stage and clip it into position. Today we are going to use the permanent slides before we make our own temporary slides. You make sure that it is in the center of field of view. Let me see how you've placed it. Alright, what's your name?
Judah Ulo.
Unafikia hiyo?
Yes.
Hebu tuchungulia tuone kama unaweza kufikia. Uko sawa. Again, look through the eyepiece while adjusting the mirror under the stage to ensure that sufficient light is passing through the specimen. Use the coarse adjustment knob to bring the low power objective lens to the lowest point, viewing through the eyepiece. Hiyo low power iko chini kabisa. Viewing through the eyepiece, turn the coarse adjustment knob gently up until the specimen comes into focus. Use the fine adjustment knob to bring the image into sharp focus. Wangapi wanaona finer details, like it becomes even more clear. Group status so far. Au wengine keep trying. Make a drawing of what you can see.
Ndio hiyo.
Yes.
Hivyo. Yeah. Unaona?
Nani hiyo? Ndio hiyo. See that one. Ndio hiyo. What you can see using the low power, eh? Yes. All right, at least your field of view is circular. For beginners, that's okay. Nani? Ilikuwa red. In biology, we do not shade, sindio? Yes, it's red, that's okay, but don't put the red colors. Do not shade. Negative? Yeah, you're still learning. Hebu let us see the red one. Hebu tuone hii red one kwa microscope. Iko sawa hapo? Kwanza hii ni nini? Hebu tuone ni nini hii yenye iko ya red. Trematoda, Faxiola hepatica, and it's there, it's red in color. Then you place it here. Your microscope is okay to observe? Okay, for starters, that's okay. You are learning how to use a what? A microscope. Even if umeona, iko wapi field of view? And this one is not in the middle. Tulisema unaweka hivyo katikati. Instead of using the low power objective lens, now you use the high power objective lens. So hii ni low power na hii ni high power. Kwani field of view size moja ilikuja kwa kidogo? Field of view, the circle is the same size, but what is the difference?
The specimen, the high power shows the specimen when they are larger, while the low power shows them when they are smaller.
Yeah, true. Iko wapi ya low power? Na hii ingine ni high power. So what's the difference between the two?
For the low power, you just see the external structures of maybe a certain object. But for the high power, it also shows you the internal details.
Oh, inaanza kuonyesha internal details. So ika zoom more. But mkumbuke tulisema tutatumia zoom. Tunatumia nini? Ina enlarge ama ina magnify more, si ndio? Good. Na ingine high power. So ni difference gani unaona?
For the low power, the image of the organisms there appears tiny.
Appears tiny, yeah.
But for the high power, it appears more clearer and more larger.
It's more larger.
Yes.
So meaning the high power magnifies more than the low power. Alafu ni image gani unaona wider part? Gani unaona wider part ya hiyo specimen? Hakuna. Ni part gani utaona wider part ya hiyo specimen? Unaona hivyo? It magnifies less. That's why you're able to see a wider part of the specimen. Bring all the microscopes up mbele. Yes, because now we are done. Have you brought all the permanent slides? All right, so we've come to the end of the lesson. Sindio? Yeah, have you learned anything new?
Yes.
What? Melan? You've learned the basics of using a microscope. So in the next lesson, we are not going to use permanent slides. We are going to make our own slides and observe them. Sindio? So now that you know the basics, it's going to be easy for you. Yes. we take the microscopes back to Biolab 1. And how do you carry the microscope?
You hold the arm
You hold the arm
and the base.
and the base to support the microscope. Sindio?
Yes.
What's your name?
Dennis.
How was the lesson?
The lesson was fantastic.
It was nice, eh?
Yes.
Aya, sawa basi. All the best. Mwarakisha mwende kwa hiyo class nyingine.